Hello viewers,
Hello everyone.
welcome to our channel Bins Biology.
So Bins is nothing but BINS. Benjamin, Emmanuel, and our two friends Nagaraj, Surya. So Bins is nothing but the acronym of our name.
Before we begin, let me ask you this. Have you ever thought about how a busy city runs smoothly? You've got roads, transports, signals, and delivery systems, making sure everything reaches the right place at the right time. Our cell works in a very similar way. Inside every eukaryotic cell, there's a whole transport network that manage a protein, lipids, and other molecules, and that's what we call endomembrane system.
So in our talk today, we'll take you through the main roots of the system: the endocytic pathway, biosynthetic pathway, and secretory pathway, including how secretion can be either continuous or controlled. And to wrap it up, I'll also share some cool methods scientists use to study the system, like autoradiography
GFP tagging.
and subcellular fractionation.
The endomembrane system is a basically group of membrane it inside the eukaryotic cell that works together to produce a modification, transports and net delivery proteins and lipids. You can think of it as a cell's delivery and recycling network. It includes in several key organelles, and the first one is endoplasmic reticulum. In endoplasmic reticulum, it have two endoplasmic reticulums, and the first one is a rough endoplasmic reticulum. That rough endoplasmic reticulum, it's starting point of the protein, and the second one is the smooth endoplasmic reticulum, and the smooth endoplasmic reticulum is the starting point of lipids. And the second one is a Golgi apparatus. It is the post office of the cell. It modifies, sorts and packs molecules into vesicles. And the third one is a vesicles. It is a delivery truck that carry a cargo to different part of the cell. Fourth one is a lysosome. It is a recycling center that breaks down a waste or unwanted materials. And the fifth one is a endosomes. It is a sorting station for material that comes in through endocytosis. And the last and final one is a plasma membrane. It is a toll gate organelle that enter or exit the substance into the cells.
Endocytic pathway. The first pathway in the endomembrane system is the endocytic pathway. It is mainly about how the cell takes in materials from the outside environment. Think of it like goods being imported into a city at the cell level. The plasma membrane folds inward and pinches off to form vesicles that brings substances inside. Now there are two main types of this process: phagocytosis and phagocytosis. Phagocytosis. It is often called as cell drinking. That's because this is when the cell takes in small droplets of fluid and dissolve substances like city absorbing water supplies. And there's phagocytosis. Phagocytosis is also called cell eating. This is when large particles like bacteria or cell debris are engulfed. You can imagine garbage trucks collecting bulky waste from the city streets. So phagocytosis is a intake of cell membrane of fluids, and the phagocytosis is intake of cell membrane of large particles like bacteria, cell debris. Okay. Once the vesicles are formed, they fuse with the early endosome, which acts like a central sorting station. From here, useful materials may be recycled back to the plasma membrane, while the rest moves deeper to the late endosome. Finally, unwanted or harmful material is sent to the lysosome, which is like the recycling and waste disposal plant of the city. So overall, the endocytic pathway make sure the cell can take in nutrients, respond to signals, and get rid of unwanted material efficiently. Okay, so you can see there are two: phagocytosis, phagocytosis. So phagocytosis, as like I already said, it is called cell drinking. So this is where the cell membrane absorbs droplets of fluids, dissolved substances, like a city absorbing water supplies. So on the other hand, there's phagocytosis. It's called cell eating. So this is when the cell membrane absorbs large particles like bacteria or cell debris. So these are the two types in the endocytic pathway. Let's talk about biosynthetic pathway. This pathway focuses on how the cell makes its own proteins and lipids, processes them and sends them to the right place. It all begins in the endoplasmic reticulum, in the rough endoplasmic reticulum actually. Proteins are synthesized with the help of ribosomes, like factories in a city producing essential goods. So in the same way, in the smooth endoplasmic reticulum, lipids are made, which are equally important building blocks. So proteins are secreted in the rough endoplasmic reticulum, and the lipids are secreted in the smooth endoplasmic reticulum. And after production, these molecules are packed into small vesicles and transported to the Golgi apparatus. The Golgi works like the city's post office and customs center. It modifies proteins, adding sugars, folding them correctly, and then labels and sorts them so they reach their correct destination. Finally, the products are loaded into new vesicles that carry them to different places. Some to organelles like lysosomes, some to the plasma membrane, and some stored for secretion. So, the biosynthetic pathway ensures that the cell can manufacture, process, and distribute all the key products it needs to function, just like city industries and supply chain. So, this is the biosynthetic pathway. So, as you can see, the left side is the rough endoplasmic reticulum, and the proteins are transported into vesicles, covered with vesicles, and it will be going to the Golgi apparatus where the protein is modified, and it goes to the various parts like plasma membrane and lysosomes and different places. Secretory pathway. Now we come to the secretory pathway, which is the system the cell uses to export its products out into the external environment. You can imagine in it like the shipping system of a city that sends out the finished goods. This pathway also begins with the protein and lipids that are processed in the endoplasmic reticulum and the Golgi, and then packed into secretory vesicles. Now, there are two types of secretions here: constitutive secretion and regulated secretion. So these two secretions will be viewed in the next slide. So the secretory pathway helps the cell communicate with its surroundings, releasing signals like hormones and maintaining balance with the external environment.
Constitutive secretion occurs continuously in all cells. Vesicles constantly delivers proteins and lipids. Maintain plasma membrane composition, which delivers extracellular proteins like collagen. It does not require an external signal. Ensure the steady flow of material out of cells. Important for normal cell maintenance. Example: fibroblast secreting collagen fibers. Regulated secretion. This occurs in specialized secretory cells. Molecules stored in secretory vesicles. Secretion triggered by external signals. Signals include hormones or nerve impulses. Allows the precise control of secretion. Timing ensures its molecules are released when needed. Is critical hormones and neurotransmitter release. Example: insulin secretion by pancreatic cells.
Next, autoradiography. Autoradiography is nothing but is a technique used to study molecular pathways by tracking molecules inside the cells. It used radioactive isotopes to label the molecules, which allows for detection. This method tracks the movement of proteins within cell, revolving pathways from the endoplasmic reticulum to the Golgi apparatus and then to the membrane. It was a classic method used by researchers like Plate. Autoradiography helps visualize the dynamics of protein trafficking, shows how proteins move within the cell. It is particularly useful for mapping biosynthetic pathways. An example application is radio-labeled amino acid tracking, which follows newly synthesized protein. Next, GFP. GFP means green fluorescent protein, derived from the jellyfish Aquaria victoria. It is used as a fluorescent marker to visualize protein in living cells. GFP is often genetically pursued to a protein of interest, enabling real-time imaging. These techniques allows researchers to track vesicles trafficking and organelle dynamics inside the living cells. GFP is non-toxic and stable, making it ideal for cell biology researchers. It revolved how cell biology researchers is conducted by enabling dynamic studies. A common example is GFP tagged acetine used in cytoskeleton studies to observe actin behavior.
Well, we move on to the next topic is subcellular fractionation. Is one of the key experimental methods used to study the endomembrane system and other organelles. Well, the process begins with homogenization. Here, the plasma membrane of the cell broken open either mechanically or chemically, so that internal compounds are released into the solution. This solution is called a cell homogenate. Once we have the homogenate, it placed into the centrifuge. That is a machine used for centrifugation, which spin the sample with very high speed. These principles are very straightforward. Organelles of different size and densities. It will be sediment or settle at a different rate when subjected to centrifugal force. At a low speed, the heaviest structure like nucleus settle at the bottom of the tube. It is called pellet. If we increase the speed a little more, the next fraction, a mitochondria and a lysosome, get separated. And the spinning at the still higher speed allow lighter organelles such as endoplasmic reticulum and Golgi apparatus to sediment. Finally, at very highest speed, we can isolate the lightest component like ribosome and the small vesicles are settled at the upper of the test tube, which is called a superdent. In simple terms, subcellular fractionation gives us a way to break down the complexity of the cell into a manageable parts. Instead of looking at the whole cell at once, a scientist can be isolated and study each organelles individually, which has been a crucial in building our knowledge of the endomembrane system.
We hope you like this video and please do like, share and subscribe.